This is a production of Cornell University.
Thank you very much for the introduction and for the invitation to be here today. I've been working with people from Cornell for a number of years, and it's really nice to actually be here in person for the first time in Ithaca, as well as actually being anywhere in person nowadays. And so it's my pleasure to be here. And so what I'm going to talk about today is some work that I've been doing over the last maybe 10 to 12 years. I'm in my research group, but before I do that, I just kind of wanted to I apologize for the sound. There doesn't mean to be sound. But I just want to talk a little bit about some of the new things my lab is doing that's really exciting. And so one of them is we just finished building this high throughput phenotyping greenhouse at the University of Illinois campus, where we're really starting to develop some new techniques and some new opportunities to advance scientific questions that I'll be talking about today. And I'm only bringing these up because I'm meeting with a lot of people and I love to talk about this stuff. And so I just wanted to kind of just give a little bit of an overview. In addition to this, my lab has just built the largest spider cam system in the world. This is a cable-based high throughput phenotyping system that gives us the opportunity to measure 4 hectares of plants within about 2 hours, generating about 20 to 30 terabytes of data on an agnostic platform where we can put anything on there. Right now we're doing hyperspectral measurements and high resolution LiDAR. I run a network of eddy covariance towers around the state of Illinois, as well as down in Florida. I don't know if any of you have ever been to Archbold Biological Research Station. I know Cornell has had strong ties with that facility, the Buck Island Ranch, for a number of decades now. And so I started doing work down there in about 2008, looking at ecosystem scale fluxes. And more recently, my lab's starting to get into the ecophysiology of integrating land uses with polyculture, both in terms of just standard, typical polyculture, as well as in agrivoltaic systems. And so throughout the next day and a half when I'm meeting with people, if you have any questions or want to talk about any of this, I'd love to do it. But what I'm going to talk about today in more depth is going to be work that I've been doing for, gosh, it's about 20 years now, but the focus of today is going to be about the last 10 or 12 years, looking at crop responses to climate change. And it's not just the responses that I'm interested in, but it's also opportunities for adaptation. And I'll get into that throughout this seminar. The Soyface Research Facility is located just south of campus at the University of Illinois. This is an aerial view where you can see, if you kind of break my neck looking up there, but you might be able to make out these different plots. Each of these plots represent either a control or an elevated, a control or a global change manipulative plot. And and what I mean by that is that each plot, 4 of these plots will have elevated CO2. Nested within those CO2 plots, we're going to have high temperature treatments, maybe some drought awnings to simulate drought. And so it's been a great research facility that's been in existence for about 20 years. But it's been something that's been adapting over time. And the impetus for this research has to do with the primary fact that CO2 concentrations in the atmosphere are rising. And this is, of course, no surprise to anybody in this room. But in addition to rising CO2 concentrations, we're seeing significant increases in other greenhouse gases, such as nitrous oxide, which are in much lower concentrations, but have a much higher greenhouse gas potential. Methane concentrations. I remember when I did my PhD, there was a lot of talk about methane stabilizing, but it hasn't. Unfortunately, it's going up. And then we also have a lot of other really potent greenhouse gas, chlorofluorocarbons and other types of refrigerants or industrial chemicals that have a substantial impact on the forcing. And what these greenhouse gases together are doing are driving global temperatures higher. And so this is just looking at northern hemisphere temperatures over the last 1,000 or so years that have been reconstructed or are based on measurements in the recent decades. And there's no doubt that the globe is warming. And the fact that CO2 is going up, right? CO2 is really important for plant physiology. CO2 is the primary molecule that's used in photosynthesis, and it's how plants grow, right? And so rising CO2 is, of course, important. And if you look at this graph of photosynthesis as a function of temperature, and you see this black line, just shifting up to this dashed line, this represents the impact that rising CO2 is likely to have on plant photosynthetic rates, particularly C3 plants, all right? Temperature, of course, all metabolic pathways in a plant are going to respond to temperature. As a result, right? Temperature is extremely important. Rising temperatures are really important for the physiology of plants. And photosynthesis is no exception. And so, of course, if you look at rising temperatures from, current at the time, this was 1991, current CO2 concentrations, it's predicted that temperatures as we get higher and higher are going to drive photosynthetic rates lower. But in this 1991 paper published by Steve Long, he discussed the synergistic effects of rising CO2 and rising temperature, leading to a much greater stimulation in photosynthesis than if you either had CO2 alone or rising temperatures alone. Of course, there are other lines on here which represent physiological adjustments that a leaf or a plant can make in response to these new growth environments. And so in elevated CO2, for example, if you get a downregulation of the primary carboxylating enzyme rubisco, you might end up with a temperature response curve that will mimic something like this dotted line or this dotted line. And so there are two parameters that are particularly important in understanding how a plant responds to growth conditions in elevated CO2 or an elevated temperature. And those are best represented on this graph here. This is photosynthesis as a function of CO2 concentration inside the leaf. And there are two different functions that are plotted here. This purple dashed line represents your RUBP regenerated photosynthesis. And then this blue line up here represents your rubisco limited photosynthesis. And the solid dark black line is what would actually be represented in a leaf photosynthesis model as your actual photosynthetic rates. And so there are two parameters that can really dictate what's happening with either your rubisco limited or our UVP regeneration limited photosynthesis. And that's VC max for this line, and it's going to be J max for this line right here. And I talk about VC max and J max because while this is a temperature versus CO2 response curve, and it's typically an ACI curve, it's typically what we use to describe VC max and J max. This is the same plot, but this is temperature on the x-axis. And so you can imagine that changing VC max or J max is going to change the way that that temperature response plot is going to behave, right? And so the general shape is going to be the same, but where that inflection between the two limiting processes or how high or how low or where the optimum is going to lie could very well shift based on how the plant adjusts to these global change conditions. And so one of the things that my lab has been interested in was trying to understand whether or not these theoretical predictions that Steve came up with in 1991 actually come to fruition if you grow plants under the real world conditions. And so as I mentioned, Soyface is a great place to do this type of experiment. And so this is one of the plots that I mentioned earlier. This is the aerial view. This is about 20 meters in diameter, and these blue pipes will emit pure carbon dioxide into the air that the turbulent nature of wind as it's blowing across the canopy will mix and carry and distribute throughout the canopy surprisingly uniformly. I mean, there's gradients, but it does a pretty good job. I was impressed back years ago, and I'm still impressed when I think about how this works. And so what my lab wanted to do was look at the CO2 by temperature interaction. And so I took a design a scaled down design that Bruce Kimball from USDA in Phoenix, Arizona came up with. And I supersized it. And so I increased the number of heaters by fourfold, giving us the opportunity to get really warm temperatures using these infrared heating arrays to simulate higher canopy temperatures in the context of these control and elevated CO2 plots. And so this would be one of the heating treatments. You can see here on the right is a view of what it looks like. And then right above this would be the reference plot. And so what I do is we measure the temperature of this reference plot up here, and then we calculate exactly how much output needs to go to these heaters to maintain some target set point. In this case, we were shooting for 3.5 degree temperature increase, but it turns out based on variation and downtime, we averaged about a 3 degree temperature increase throughout the season. And so this is what some of the data looks like. This is canopy temperature as a function of time of day. And what we have here are two different lines. They're hard to distinguish. There's a bunch of blue dots and a bunch of black dots, and they're pretty much on top of each other. The black dots are your control CO2 non-heated plots. The blue are elevated CO2 plots without heating. And then we have our two heated plots. The red, which you can barely make out, is just heating. And the green is your heating with elevated CO2. And so this is just an example from one year, 2009. And it's showing you that the difference between the black and the red line is about 3 degrees Celsius, and the difference between the blue and the green line is also about 3 degrees Celsius. And so one of the things we wanted to understand, okay, well, yeah, we're increasing the temperature of these, but we're increasing the temperature relative to what's happening in the reference plot. What's happening in the reference plot is going to change from year to year based on just natural variation from growing season to growing season. And so what I'm showing here is just a real simplification of what's going on with precipitation in the gray bars, and what's going on with your minimum, maximum, and daily average temperature for, this is just 5 years of data I'm showing right now, the first phase of this experiment. I mean, I'm showing these relative to your long-term means. And so you can see that 3 of the years we were wetter, and actually these are growing season precipitation and growing season temperatures, not yearly. And then you can see that there doesn't seem to be a lot of variation with temperature, but in fact, 2009 and 2014 were relatively cool years. We see that maximum temperatures are really where you're driving things much higher, the higher end of those error bars. And so we saw quite a bit of variation. And so the first phase of this experiment, we really just wanted to know, are these plants responding as Steve Long in 1991 predicted? And so this is daily integrated photosynthesis measured with gas exchange, just a bunch of people taking Lycor 6400s out there in the field, clamping on the leaves, and measuring photosynthesis as much as we possibly can from sunrise to sunset. And what we saw was that, as predicted from Long 1991, in this first year, we saw a slight decrease in photosynthesis in elevated temperature and a slight increase in elevated CO2, and not statistically significant, but maybe a little nudge higher in the elevated temperature plus elevated CO2. And this is consistent with kind of the predictions that Long 1991 predicted. But then in 2011, which was a much drier and a much warmer year, we actually saw that the Long 1991 predictions did not hold up. And the fact that we saw substantial decreases in photosynthesis in high temperature, we saw increases in photosynthesis in high CO2, but we did not see this synergistic effect. In fact, this elevated temperature plus CO2 had lower photosynthetic rates than the elevated CO2 by itself. And in fact, we did this over 5 years. These are 2 different papers. We saw the same response occur with regard to seed yield. Although there wasn't an increase in seed yield in 2009 in elevated CO2, it kind of follows the same pattern we saw with photosynthesis. This pattern, actually, the decline in yield for this combined treatment is even greater relative to the CO2 plot than the decline in photosynthesis. And if you just look at these white bars down here, these are 3 additional years of data showing you that most years are actually kind of following what we're seeing with 2011. And so in fact, most years we're seeing this CO2 plus temperature effect not doing really well. In fact, marginally, no different than the elevated temperature alone, all right? And so this suggests that the synergistic effect of elevated CO2 and elevated temperature actually doesn't hold. And so we're wondering why. 2 things. We're wondering why isn't this theoretical? I mean, mechanistically, using the Farquhar model, the modeling that was done in 1991, that's what we understand of the mechanism, but we're not seeing that with photosynthesis. And we're also seeing larger declines in yield than we are seeing declines in photosynthesis in the high CO2 plus high temperature treatment. And so it turns out, this was the graph I showed earlier, the control graph, like our ability to control the temperature. You can see there's not a big difference between the canopy temperature for the control and elevated CO2 without or with heating. Most years we actually see something that looks like this, where in the middle of the day, there's a clear difference. Your control plots, low CO2, no heating, is the lowest temperature. Elevated CO2 plots, we didn't add heating to these plots, the blue. This is just the fact that elevated CO2 reduces stomatal conductance. That reduced stomatal conductance warms the plant canopy. The red, this is 3 degrees temperature increase above the black, and then the green is a 3-degree temperature increase above the blue. And so what we suspect is happening is one of the things is that the higher temperature in the elevated CO2 plus elevated heating, basically that elevated CO2 induced stomatal closure is probably bringing more temperature stress to the plant canopies than the heating treatment alone. And so we're actually doing 2 heating treatments. The first heating treatment is the infrared heating array. The second heating treatment is the closing stomatal aperture because of elevated CO2. And in fact, if you look at all the other years, like I said, I didn't put the graph up there, we see something that looks very similar to this. All right. And so we actually have 2 separate heating treatments that are being imposed, one through the infrared heaters, one through just the increase in CO2. But that's not the only story. I mentioned photosynthetic acclimation. When you grow a plant under certain environmental conditions, you end up with a value for VC max and a value for J max. When you change the growth conditions, the growing conditions, VC max and J max are likely to change. And this is one of the big challenging aspects of modeling, whether it be at the leaf, the canopy, or the ecosystem, or the global scale, is trying to understand how these physiological parameters, which are really important to driving the carbon dynamics of an ecosystem, it's understanding how these parameters change. And so from many years of research, I have more than this, but we're seeing a very consistent response in that with VC max, which again is that initial portion of the photosynthetic CO2 response curve, we don't see a whole lot of difference in the VC max for plants grown in control versus elevated temperature, so the black versus the red bars. In elevated CO2, we do see significant declines in VC max, whether or not heating is given. So this heating treatment doesn't have a big impact on VC max, independent of what your CO2 treatment is. But CO2 treatment does lower VC max. J max, we generally see a different story, which is that heating tends to decrease J max, whereas elevated CO2 has a marginally small impact on J max, although it's still significant and it's still there. And so the fact that we're seeing these adjustments in VC max and J max are actually driving slight shifts in the photosynthetic temperature response curve, as shown here. Again, elevated temperature drives photosynthesis lower. The dots represent your temperature optimum of photosynthesis. Elevated CO2 increases the temperature optimum, and it moves the optimum, increases your absolute rate of photosynthesis, and it moves your temperature optimum to a higher temperature. Elevated temperature plus elevated CO2, we're seeing a decline in your photosynthetic rate and maybe a slight increase in your thermal optimum, all right? And so these processes, this physiological adjustment is likely one of the main drivers, at least at the leaf level, for our observed responses of photosynthesis. So one of the things that I was interested then is trying to figure out what does this mean for canopy level photosynthetic rates. And so to do this, I relied on a collaboration I had with Darren Drury, who was working for Praveen Kumar at the University of Illinois in the Department of Civil Engineering, and then he went on to JPL, and I think he's at Michigan State now. He developed this MLCAN model, which is a really nice model to look at canopy level carbon and energy dynamics. This is not an ecosystem model at the time, it was just a canopy model. And he developed it for corn and soybean. And it incorporates all of the mechanistic understanding that we have of various different modules that lead to photosynthetic carbon uptake and energy balance of what's happening. It breaks the canopy up into 15 different layers into sunlit and shaded fractions. And it does a really good job, and it's been validated against eddy covariance data that my lab has been collecting at the University of Illinois in both corn and soybean. And so we figured it's been parameterized and validated based on data from our eddy covariance sites. Let's apply this to the temperature work that we've been doing. And so we took the VC max and the J max values and all the physiological measurements we needed in order to parameterize this model for each treatment independently. And this is just showing you a visual representation for 2009. This is soil level all the way to the top of the canopy. And this is from midnight to midnight. And the colors represent photosynthesis. And so keep in mind that these don't seem like they're very high photosynthetic rates, and they aren't. But you have to integrate all 15 layers at any given point in time to get what the canopy photosynthetic rate will be. And so we did this for 2009 and 2011. And then we did the exact same thing, but for the measured parameters for your elevated CO2, elevated temperature, and your combined elevated CO2 plus elevated temperature for both years. And you can kind of see that maybe the intensity of the colors are shifting based on each of these treatments, but here are just the difference plots between the treatment minus the control. And so what we're seeing is that the biggest differences we're seeing in your canopy photosynthetic rate are that we're seeing higher photosynthetic rates in the high CO2 treatments. We're seeing decreases in your photosynthetic rate throughout the canopy in the high temperature treatment. And in fact, it appears, and it's generally so, that this represents sort of an additive effect of this minus this. And so, and we see that again for both years, although the differences in the heating treatment here, the decline is greater as the model predicted, as matched with the measurements that we had from the field. And so then we wanted to ask the question, all right, we have this physiological adjustment that's occurring. VC max and J max are changing. Does that really matter? If we just ran this model, given the variation throughout the growing season, if we just ignore the acclimation responses of VC max and J max, can we just run the model and still get reasonable results? And so this is actually doing the exact same thing. We're changing the model forcing. And so the temperature and the CO₂ data is there, but we've just held all the physiological parameters constant. And what we see here again is the difference plot. In this case, it's what we got with or without acclimation. And so here you could see that if we ignore acclimation in elevated CO₂, we're slightly overestimating your photosynthetic rate. If you ignore acclimation in elevated temperature, I'm sorry, I have this the other way around. But if you ignore acclimation in elevated temperature, you're grossly overestimating your photosynthetic carbon uptake at the canopy level. And we saw this consistent for both years. And this is just a way to really summarize that. And so this is your leaf level, daily integrated photosynthesis, and canopy level, daily integrated photosynthesis. This top row is our control. And then we have our elevated CO₂ with and without acclimation. Not a big difference here or here. Here's our elevated temperature with and without acclimation. Now we're starting to see a pretty big difference, right? So you're basically over estimating your photosynthetic carbon uptake if you're not adjusting for the physiological responses of photosynthesis. And when you look at the temperature and CO₂ combined treatments, it's the same story. You're overestimating things if you ignore the acclimation responses. And so not only is this acclimation happening, not only is it having an impact at the leaf scale, and we predict that it's having an impact on the final yields of these plants, but when you take the mechanistic understanding of what's happening with these systems and you plug it into a model and you ignore acclimation, you're actually probably okay in elevated CO₂, right? Acclimation probably doesn't make a big difference in elevated CO₂. And there's reasons for that, that mechanistically make sense. But once you get into elevated temperatures and you ignore that acclimation response, then your model's not going to match up with what we're measuring in the field. I want to point out, this is very specific to soybean. Soybean, we see that there's this decline in VC max, but no change in J max. And as CO₂ goes higher and higher, you're more likely to be J max limited. And so losses in VC max are not a big deal, right? But not all species are the same. And so if you look at certain grasses and shrubs and trees, well, these models predict that you're going to be limited by VC max, both under current and future CO₂ concentrations. And so ignoring VC max in those models, in those systems, you might actually be grossly overestimating what's happening. And so keep in mind, this is very agriculture soybean C3 focus right now. But there's a lot more that needs to be done to understand what's going on with these other systems. So I wanted to point out that it's not just about yields, right? I mean, Illinois, we live, breathe, and die. We make a joke. We have both kinds of plants, corn and soybean, right? And, you know, I mean, walking up the steps here today, seeing all these different shaped leaves are like, wow, crazy. But it's not just yield for me. I mean, there's a lot of other facets of my research that I find. In fact, I'm not even a huge fan of corn and soybean, to be honest with you. Don't tell Illinois that. But it's also quality. Nutritional quality is extremely important. And I know most corn and soybean don't go to feed humans, but nevertheless, nutritional quality is important. And so a few years ago, there was a study that was published. I'm not part of this, but it was showing you that in elevated CO₂, for a number of different types of crops, you're seeing a decline in the nutritional quality of the final product. And so we're curious whether or not our experiment showed similar types of responses. And so this is just showing you a number of different elements of the yield. This is work that I did in collaboration with Ivan Baxter at Danforth. This is our previous reports from other studies showing consistent declines in a lot of the various different nutritional components of the yield. And just for 2 of the years of our studies, we see that, you know, either we see no difference or we see declines in elevated CO₂. And so we're seeing either no change in nutritional quality or we're seeing a decline. We're not seeing any improvements in quality in these soybean plants going in elevated CO₂. But when you look at the temperature effects, we actually see that while yields tend to go down, the quality of that yield tends to go up. And so where there are changes here, with the exception of phosphorus, where there are changes, they're almost always to a higher nutritional quality. And so while there's a lot of work showing that elevated CO₂ drives nutritional quality down, elevated temperature tends to improve the nutritional quality. And when you grow them together, which isn't shown here, they tend to offset each other. And so you end up with no real big change in yield, and you end up, or a potential decline in yield, and you end up with no real big change in nutritional quality. We also looked at oil and protein content, and we actually, this was just done at different layers in the canopy, but we saw some mixed results here. And so the oil percentage oil of the soybean went up in elevated temperature, but didn't change in elevated CO2. And the amount of protein as a consequence went down in 1 year. We saw generally similar results, but a lot more variation in 2015, and we're not entirely sure why. And so more research is in the process. We're just waiting for Ivan to do the analysis on the yields. So everything I've talked about right now is focusing on elevated CO2 and global warming. And I've been talking about global warming in the context of just mean increases in global temperature. Fact is, and that's what this is showing here. This is probability plots of temperature. The average is here, and we're predicting the average is shifting to here. And we're simulating 3 degrees Celsius in the experiments that I've been presenting thus far. But we can also end up with an increase in variance in temperature. And that's what's shown in this middle figure here, where the actual mean temperature doesn't change, but you're seeing that you're pushing down on the probability plot. And so the low and the high extremes balloon upward a little bit. And so that's an increase in variance. If you actually add these together, we end up with an increase in mean temperature as well as an increased probability of high temperature extreme events. And if you actually look at data that's been collected over the last few decades, this was from the 1950 to 60. As we move down to the most recent decade from 2011 to 2020, you'll see that we're seeing a shift to the right, global warming, but we're also seeing the ballooning on the high temperature extremes. And so we're seeing both an increase in mean temperatures as well as an increase in variance. And that means a warmer planet with much higher probability of extremes. And so one of the things that I did when I first set up this T-face experiment in 2008 was I set up the prototype, I turned it on, and I was excited. So I went to lunch. And I came back and realized that I got a decimal place wrong. And instead of hitting about a 3 degree temperature increase, it was up to 18 degrees Celsius. And I killed the prototype plants. They all died. And I realized now that, well, we could actually get up to 18 degree temperature increases, and that's not advisable. But after we got done with this first phase of the experiment, I thought to myself, like, let's simulate heat waves because they're becoming more common and they're probably going to continue to become even more common in the future. And so we designed this experiment where we wanted to impose a heat wave, but we wanted to make sure that the heat wave we imposed is not actually just kind of like raising the temperature to an optimum, but rather raising the temperature to an extreme. And so what I did was I looked at the long-term means, and I came up with this temperature, the diurnal temperature response, which would be the blue line, of what would happen on an average day in Illinois on this day, right? And so that's the blue line. This is our baseline, right? And then we wanted to say, okay, how much of a temperature increase do we want to give? I'm like, well, let's do 6 degrees. I did an analysis and showed that 6 degree temperature heat wave in the summer is statistically probable enough that we should, that there was a threshold. There was a threshold that seemed plausible at the time, although I would probably argue it should be higher now. This was 10 years ago, 8 years ago. And so that would be the red line. And this red line, that would be 6 degrees temperature increase. And so the goal was to increase the temperature to 6 degrees above whatever is happening on that day, but always to get above the mean temperature for that day. And I just made a mistake and I realized that. This black line is your 30-year mean, right? And so what we're shooting for is our treatment actually wants to be this blue line or higher. So we want to impose a heat wave that's 6 degrees Celsius warmer than the average temperature, at least, all right? And so what I mean by that is on this particular day, you'll see these black dots. This represents the actual canopy temperature measured in our reference plot. We got down to 12 degrees Celsius on this day. So if we increase this temperature by 6 degrees, we're going to be at 18 degrees, which is here, which is kind of ideal, right? That's not really a temperature stress. And so in this situation, we wanted to increase the temperature at least to this blue line. And there's a lot of variation here, but that's what we did on average. And then as the day progresses, all these lines are shifting because, diurnal fluctuations in temperature. And we did not want to go above this red line because that's the death zone. We did not want to kill these plants. We wanted to stress them, but we didn't want to kill them. And so we tried to never go above this. You might be asking yourself, why this variation? I like this day because it shows you the really cool temperatures, a lot of wind, where we lost the ability to control the temperature control. All of a sudden, the temperature started to increase because the front finish passing through. Now we see that the variation in the actual measure, our control, actually tightens up, and then we have this night where it was really still, and we see the upper threshold of how good we can control the temperature. And so this is showing you the extremes of what we can get. Higher temperatures of the canopy with a lot of variation, higher temperatures of the canopy with actually practically no variation. And this is all just driven by how fast the wind is moving that heat away from the plant canopy. And so we have these four different plots. Control never got a heat wave. Plots 1 got a heat wave early in vegetative development. And plots 2 and 3 got heat waves in reproductive development, early and late reproductive development. Each plot only got one artificially imposed heat wave. And we replicated that 3 times. And so this is kind of what it looks like, the red and the black line, and we matched the infrared radiometers, and then we turned on the heat wave, and then we're looking at these red lines, the heated, the black line is the non-heated. And what we see is that this is just, there's a lot going on here, but this is showing you measurements at the leaf level for temperature, photosynthesis, stomatal conductance, and quantum efficiency of photosystem 2. And this is for 3 heat waves in the first year of the experiment, and then 2 heat waves, vegetative and reproductive, in the second phase or second year of this experiment. And I want to point out that there's a lot in this graph, but ultimately what we're seeing is that there is definitely physiological responses of these plants to the heat waves that we imposed, right? And that's the divergence of the red and the black lines. Sometimes it's greater than others. In almost every situation where we saw a response, it was going to either be warmer temperature or lower photosynthesis, lower stomatal conductance. And generally speaking, not a whole lot with quantum efficiency of photosystem 2, except maybe on 1 or 2 days. But because no matter when we gave these heat waves, we were imposing a strong physiological response to the plants. When you actually look at the impact of these heat waves on final yield, however, all of our results suggest that a heat wave during vegetative growth of soybean has no impact on the final yield. So the plants are able to recover. But any heat wave that we impose on these plants during reproductive growth, whether it was early or late in reproductive growth, we ended up seeing a decline in your final yields. And so all the treatments saw physiological responses, but only in the reproductive phase of this experiment do they actually translate to decreases in final yield. And then, of course, we wanted to take a next step to see whether or not CO₂ has an impact on this. And these are just another 2 experiments that we did in 2015 and 2016. Here we did a 9-degree temperature increase. And again, this was a postdoc that made a mistake. Didn't kill the plants, but boy, did she really heat them up, and which was great. What we see is that these really high temperature treatments, it seems that the elevated CO₂ plants, heating plus elevated CO₂ plants saw a greater decline in yield than when we only heated by about 5 degrees. And so it suggests that another layer of complexity is not only does it matter when the heat wave is going to hit, but the intensity of the heat wave is also a major factor. But it seems like the elevated CO₂ with the heat wave, the elevated CO₂ doesn't seem to provide a great deal of protection against the heat waves. And so from our analysis, at least with soybean, it would suggest that as these heat waves become more and more common, the rise in CO₂, despite the increase in productivity associated with elevated CO₂, may not necessarily provide any sort of protection against the heat waves that we're likely to continue experiencing. And so this also shows up, at least in 2015, where we saw the huge impact. We saw final yield levels are down regardless of whether or not CO₂ is present. But in the lower year, we actually saw, this is the first year where we saw this really significant increase in this one scenario in yield. And so there is this threshold idea, right? And so how much of a heat wave is really going to be enough for that tipping point to be reached? That's something that we would like to get at, but and hopefully we will at some point. This is an evolving experimental facility. But before we get there, one of the things that really came up that were of interest is recently there was a project funded, not recent, I guess it's been 10 years now. COVID throws time off, I don't know. But it's this project to try to improve photosynthetic efficiency of crops. And it's the Realizing Increased Photosynthetic Efficiency project at the University of Illinois. And the goal is to try to overcome the fact that for a lot of our crops, particularly the C3 crops, we're seeing yield increases per decade are starting to decline. In fact, they have been in decline for some time. And so new techniques, new methods, and new opportunities need to be sought in order to realize increases in yield that extend beyond what traditional breeding has been able to do. And so some of the work, these genetic modifications that I'm going to be talking about are not work that I do. I'm a physiologist, mostly environmental physiologist, but I work with people who do a lot of genetic modifications of plants. And so Steve Long's group, in collaboration with Christine Rains at the University of Essex, they did a modeling analysis to try to optimize the Calvin cycle enzymes and see, are they optimal for plants grown in today's atmosphere, and how do they need to be optimized for the future environmental conditions. And in this optimization strategy, which I'm not going to get too much into depth with, they identified that in order to optimize cellulose bisphosphatase, one of the 9 Calvin cycle enzymes, for future conditions, you should actually have almost an order of magnitude higher concentrations of SBPAs. And so there's right now it's suboptimal. And the theory that Steve came up with when he did this was that it's suboptimal because plants evolved in lower CO2 than they're experiencing right now. And CO2 concentrations have gone up quite substantially in a short period of time, and the plants haven't been able to keep up. And another strategy that is used to try to improve photosynthetic efficiency is a photorespiratory bypass. And so C3 plants will actually assimilate oxygen in oxygenase activity of rubisco and drives this photorespiratory pathway that requires quite a bit of energy, loss of carbon dioxide, and shuttling of carbon amongst 3 different organelles. And so the idea is to try to come up with a bypass where this phosphoglycolate that's produced from this oxygenation can just be processed here in the chloroplast with less energy, and the CO2 is still going to be released, but at least it's being released around rubisco, where photosynthesis, the primary entry point of carbon into the plant. And so these are 2 strategies that we considered might actually be pretty good with regards to resilience to high temperature. And so that's because this is a graph from earlier showing that at high temperature, Jmax tends to go down, right? And SBPase is one of the enzymes that's responsible for regenerating RUBP. And so in theory, if we see an elevated temperature, that this black line here goes down to this dashed line because of photosynthetic acclimation, can we just try to manipulate this dashed line to come back up here? And we thought SBPase seems like it might be a strategy to do that. And so these are the SBPase overexpressing lines of tobacco that Christine Rains grew, are developed at the University of Essex. And it's showing you under just control conditions in a greenhouse or growth chamber, your wild type plants are smaller than your SBPase overexpressing lines. And she also showed that by measuring CO2, photosynthetic CO2 response curves, these SBPase overexpressors are actually looking at higher Jmax values, right? And so it seems like this would work well in high temperature. And so we actually did grow, we actually got SBPase overexpression in soybean, all right? We actually work a lot with tobacco in Illinois, but obviously I'm not a huge fan of corn and soybean. I'm even less of a fan of tobacco, but it's a great model crop species. And so it's fun to work with, but that's it. And so we got this into soybean because that's really what we're interested in. And this was in collaboration with Steve Long. And so we grew both of these in the temperature experiment out at Soyface. And what we basically saw was that we actually saw slight increases in the SBPase overexpressors, both in terms of VCmax and in Jmax. And so that was a bit surprising, seeing this VCmax response, but the Jmax response was something that we predicted, and for 3 different years of experimentation, we saw that. And we also saw that photosynthetic rates in the wild type versus the SBPase overexpressors, and this is for all treatments, has actually gone up in most of the, so this is morning, midday, and afternoon for 3 different years. Mostly in the midday and the afternoon is where we saw the greatest increases in temperature. And it turns out that the higher the light and the higher the temperature, the greater the stimulation in photosynthesis as a result of the SBPase overexpressing lines. But the big question we had was, what does this mean for the temperature treatments or the CO2 treatments? And so what we saw actually surprised us, which is that for the control, elevated temperature, and elevated CO2 treatments, there were no differences that we can observe in final yield between the control and the wild type and the SBPase overexpressor. And this was for all 3 years, with the exception of this one control anomaly that we had in the 3rd year. What we also saw, and this was, I showed you this earlier, I said ignore the gray bars, now don't ignore the gray bars. What we're seeing is that the SBPase overexpressors in all 3 years actually had higher yield in the CO2 plus temperature treatment. And so it seems as if SBPase overexpression doesn't lead to higher yields in high CO2 and high temperature, but it does protect the plants, the final yield, against the detrimental impacts of higher temperature. And we're not entirely sure why, except that this was observed 3 years in a row. And so there's a lot of questions, but it suggests an opportunity for adaptation to high temperatures. And just briefly, I'm going to just present some of the work on the photorespiratory bypass. And so this is, again, work that I've done with a collaborator. This time it's Don Ort and a postdoc in his group, Amanda Kavanaugh, where they developed these. Not only did they have to come up with this pathway in the chloroplast to metabolize the phosphoglycolate, but they also had to actually cut out a transporter that competes with that phosphoglycolate being transported out of the chloroplast. They actually had to stop the export of the glycolate from the chloroplast. And so this was, this alone doesn't do anything. You have to actually knock out one transporter. And so we got our hands on these and we grew them out at, this time it's tobacco, right? Not funded by Philip Morris, I promise. And we did, in 1 year, we imposed 2 different life cycles. Tobacco has a pretty short life cycle in Illinois. And so we grew 2 different cohorts of plants, an early season experiment that was harvested and analyzed, and then a later season experiment. And so there are 2 different heat waves that we imposed on 2 different lines, 2 different separate experiments. And we're shooting for a 5-degree temperature increase in these, sorry, it's not a heat wave. We grew them the whole life cycle. But so this is the entire lifespan for tobacco early season and the entire lifespan of tobacco for the late season. You know, and what we saw is that the photorespiratory bypass had higher photosynthetic rates. Their thermal optimums seemed to shift to higher temperatures. And that occurred despite not a whole lot of change in your VC max or your J max. And so it suggested that there's not a whole lot of photosynthetic acclimation going on here, but this photorespiratory bypass is yielding slightly higher photosynthetic rates. What we did see, however, is that the wild type plants, we saw a decrease in above ground dry biomass in both the wild type and the photorespiratory bypass lines. And we saw this for both experiments. But if you actually look at the wild type biomass decreased by about 35% in elevated temperature, whereas the photorespiratory bypass also decreased in high temperature, but they only decreased by about 15 to 18%. And so again, it's one of these ideas where implementing this photorespiratory bypass can potentially increase biomass. We don't know about yields because this is tobacco, right? The seeds are ridiculously small and no one wants to harvest those anyway. This is just biomass. But what we see is that this photorespiratory bypass does not result in higher ability to adapt to high. It doesn't increase your photosynthesis or your biomass in elevated temperature, but it does protect against some of that loss in higher temperature. I'm just going to finish with something we just started. And so for at least 2 or 3 of you, based on my experiences in talking about this work, are probably sitting there thinking to yourselves throughout this entire seminar, you know, Carl, when you're increasing the canopy temperature, aren't you also messing with the vapor pressure deficit? All right. And answer is absolutely. And so as we use these infrared heaters and we shine them on the plants, right, we're increasing the canopy temperature, but we're also changing the leaf to air vapor pressure deficit. VPD is a really important parameter for how plants grow, right? It dictates how much water is being lost to the plant and has this impact on the stomatum. And so what we've done, what my lab's done, I've been working the last 1.5 years to build the system, which we just built and it's been out in the field. This was actually from last year. This is just a prototype. Where what we're doing is we're increasing the canopy temperature as I've been talking about through this whole seminar now. But in addition to that, I built a misting system that atomizes water vapor and will actually inject water vapor into the air to increase the atmospheric humidity at the same time that we're increasing the canopy temperature. And so this is just showing you some of the control. This is our control. This is the mean and the just box plots of VPD. And then the elevated temperature treatments, you can see that this VPD is shifting to much higher values. And that's because we're artificially increasing canopy temperature. This first year, we applied the relative the water vapor, but nevertheless, we're seeing really strong. This is high temperature plus the additional atmospheric humidity. And we're seeing that our VPD is much lower than the control. So we actually have 3 different VPD treatments here and 2 different temperature treatments. Our goal this year, which we should get the data pretty soon, was to try to match this and this as closely as we possibly can. And I don't know if we've done it, but it looks promising so far. And what this is going to do is I have postdoc Nick DeLucia, who some of you might know his dad, Evan DeLucia. He's been out there making a ton of measurements on these plants throughout their entire life cycle to try to understand what are the physiological implications, particularly related to photosynthesis, of the elevated temperature versus the elevated temperature plus the higher VPD treatments relative to the control. And for those of you who do any sort of ecosystem modeling, that's one of the hardest things to get at right now is understanding the temperature versus VPD effect, because they tend to change together almost universally. So, all right, that was a lot. I hope I didn't go over and maybe went under. I guess I talked last time I gave this, so I went over. Before I finish, I do have to acknowledge the fact that everything I've done on this is a collaboration between me and postdoctoral and grad researchers and the grad students in my lab, as well as collaborations with a lot of my colleagues, including Don Ort, Steve Long, and the postdocs and graduate students in their lab. So this is like collaboration with a capital C, and I need to acknowledge them and all the people that have contributed to the various different aspects of this research. So with that, I'm happy to answer any questions you may have, and I appreciate your attention. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.